Hi, everyone. I'm Franco Pestilli from the University of Texas, Austin. It was a wonderful day. First of all, I'd like to thank all the speakers for the day. I'd like to thank Tui for the wonderful moderation work that she has done. So now I recognize how hard it is to see you guys. I have a question for you. How many of you do not work on any of the tools that we've been seeing today? Can you please raise your hand? How many of you do not work developing these tools?
What, not using or not developing?
Okay. How many of you work developing the tools that we have seen today? Okay, very small number. Okay, so hard. The not is hard. So how many of you work and think about yourself as a clinical neuroscientist? Okay, a few. How many of you are cognitive neuroscientists? Okay. How many of you are informaticians, work in informatics? All right, good. That's just to calibrate the questions that we have. Maybe we should have done that earlier today. But it looks like we have a good room of people that actually are interested in this question. If I were to make a summary of what I heard today that we don't have in neuroimaging one way to do it right. Okay? And in what we have many ways to do things that approximate what we believe is pretty okay. So, and now there's a series of tools, statistical tools and informatics tools, thank you, that we've been developing as a community that help you guys, now I can say that, you guys, to figure out, a consensus approach, right? So I think Tom's talk at the end of the session really wrapped up some of this idea, generalizing to what is the fundamental statistics here, right? So I love the plot where you can see that there's a consensus, it's an agreement across all of the different models, and there's some differences, and how do you evaluate what is common variance and what is not common variance, and what you should believe and what you shouldn't believe. Now, there's a lot of tools that you can use to do what is really multiverse analysis, right? And which tool should you pick depends on how you like to do it, right? Do you like interfaces? Do you like to use this tool because you're already familiar with it, so you will add on top of it another one that is very similar to it, or whether you do want to start from scratch, et cetera, et cetera. Now, one thing that seems really hard is that we're adding a layer to the complexity. We have a complexity which is we don't have one solution that for what is the best way of analyzing data. So the solution is do it many times. And in addition to that, we're giving you all these different tools to do it many times, right? So now there's a multiverse analysis of tools, and you can do a multiverse analysis of the tools that let you do the multiverse analysis of the tools. All right, I'll stop. So, and now I go to some of the questions that we prepare for the panelists. How can we ensure that the entire research process from acquiring the data to processing it, not just the data, is reproducible? I'll start with Satra with this one, because I know he likes to think about this stuff. How can we ensure that the entire research process, not just the acquisition of the data or the processing, is reproducible?
The short answer, I don't know if we can. And I'll start with when I was a grad student, FreeSurfer was in its infancy. And you wouldn't get the surfaces that you get from FreeSurfer now. You had to hand edit every brain to then make FreeSurfer work. So in many places, and I'm going to extend this thought process a little bit, we had to kind of intervene to make things work, to produce an output that we could trust to move the next step forward. Today, some of those steps have been taken over by algorithms. And tomorrow, these might be taken over by vision language models and other things that can evaluate the output, as Stefan was showing, for example. Oh, I see a piece of the brain. Can I tweak the AFNI parameters so that I automatically improve this? So this leads me to this notion that what is reproducible analysis if we start applying different strategies to fixing up parts of the pipeline in different ways, from data collection to evaluation at the end of the day? Maybe I'll leave that for a question for the rest of things. My short answer is, I don't know if we can.
And I would just reframe the question, which I think just asking how to make it reproducible, and that makes it sound like it's a binary thing. And rather, it's like, what do we do to make it as reproducible as possible? I mean, let's be realistic.
Anyone else wants to pitch in? JB?
Actually, I will take that as a segue for something that just before that session, there was the CSIG session on carbon footprint of those processing. So Nikhil organized that, and also actually participated in the questions. I think you was the one putting the questions to the panel. And I'm just struck by the fact that the tension between our goal in science to try to get to the results that are valid and correct, and at the same time, if that means that we have to use 10 pipelines to have a good idea of how those pipelines, what kind of results you get, that is going to increase the footprint, carbon footprint of things that are. So I'm just trying to think of how do we correct ourselves and basically say, oh, with that kind of data, this is the pipeline that is reasonable, and that would mean a data sharing and a pipeline sharing, and some information on that level that would be much more than what we have at the moment. So I'm just thinking globally at that problem is really we have to think differently, I think, as a community, because if I knew that specific brand, for instance, that you were looking at, Stefan, doesn't work with the FNI pipeline with those parameters, if I had thousands of those information, then maybe there's something that can be done to say, okay, look at that brand, and then now decide what kind of pipeline I can use, and reducing the carbon footprint that is a problem, is a real problem.
Yes, and I fully agree. I just want to add to this. I think there are two general things that I always try to keep in mind. The first one is that all of these pipelines, because people often ask me, what's the correct pipeline to run? Right? And my answer is, there is no correct pipeline. All of these pipelines are models, and as we know from HBox, all models are wrong, some are useful, right? And this is the true thing. Some of these, and that's the question, what is useful? And I think this is where we have to zoom out again. What are we actually trying to answer here? And I think there is probably a good enough way of doing it. There's a good enough way of enough reproducibility to get what you need. But I think this changes depending on the question. And I think this is something then where we as humans come into the game and need to make that decision. And I think that also comes into, okay, how complex does my pipeline need to be? Because if I look for something quite simple, I don't need a fancy pipeline sometimes. Yes. So I think keeping it simple, Occam's razor, we're modeling, and we need to look at the question that we're actually trying to answer.
I think there's a question or comment from the public.
Yeah, so I think what we are seeing here is we're a bit in the intersection between reproducibility and methodological variability, right? So one of the earliest papers talking about this is these two pair papers by Joshua Karp back in 2012, something like that. And there is some message there that I think is usually lost when we talk about these things. And basically what he was proposing is methodological variability is not that dangerous up to the moment where you don't report what you did. So basically, one of the big problems with reproducibility is that either because we're not aware of the complexity of what we are doing, we don't report the whole space of parameters we have tried to search, or because we tried a lot of things and we only report the one that worked for us. In the end, that's what makes things irreproducible, because then I tell you I did this, you try to do it, and you say, no, it doesn't work, right? So I wanted to ask you all, as experts, one thing that in the NARP's paper is proposed as an alternative to the multiverse analysis, which is the pre-registration.
Well, it's the p-hacking as a pipeline hacking problem, right? Yeah.
So could be pre-registration a way of at least committing to make your analysis in a single way. And if you couple it with register reports in publishing, making sure we don't try to fine-tune until things work as we wish.
Can't say anything against pre-registration. Yeah.
Maybe Stephen, do you want to?
Can we pass the idea of reproducibility here, because we've got the reproducibility of the software, which we all know about, but you don't actually want your results to just be reproducible, because a highly biased result is perfectly reproducible. So there's actually something else that we need to worry about, and I think there's this implicit assumption that most of the choices that we make with our pipelines are going to get us somewhere into the general vicinity. But it's that bias that actually is a critical piece of the whole problem. And so reproducibility by itself, we have to remember, is not enough. Pipeline that produces one for everything is going to be perfectly reproducible, but totally useless because it's massively biased. So we've got a little bit of something here that we're sort of, I don't know, tiptoeing around in some sense. So the question is, how do we also get some sense of the bias in association with our multiverse of solutions?
I mean, and just to make that concrete, if you are someone who thinks that it is a crime to use global signal regression, that's a perfect example of doing it makes it very reproducible in one way, but then you probably think that's the wrong way.
Right. I mean, I've published some possible ways of going about that, but I'm not saying I have the answer. But I think it's very important to remember that reproducibility is just a piece of the thing we're interested in. And also you've got to make that distinction between the software reproducibility, which we probably don't have because we end up dropping these containers on top of libraries in OSs, which we can't be absolutely certain about, well, at least as I understand it, not yet.
Yeah, I mean, I can just add to that. I mean, we tested this quite a bit with containers. So the cool thing is with containers, you actually have a lot of the libraries that you depend on in the container. So glibc, for example, which a lot of variability would come from otherwise. What we do see as a leftover residual is hardware effects. So for example, you run on an AMD versus an Intel versus an ARM processor. But these are like an order of magnitude lower. So I think by putting it in a container, we're like a very far step into this world of, okay, now we actually, first we know what we're running, which is very important when we publish this. And then it is pretty reproducible.
Is that also true if you're doing sort of SVD decompositions, which can tend to be a little unstable?
Well, exactly. So I think if we say like it's library dependent and software, we trace these things through. I mean, TUI did a lot of tracing of these libraries. We know that we're taking the same execution paths. We also know that this is not enough. There is then still floating point precision underneath that we have absolutely no idea and no control about, except we're running on the same processor. Still, even if you control that library, there can be switches in these libraries where you actually change the execution path based on the hardware that's underneath, and then you're back into this, okay, now I'm actually doing different things. But this is definitely lower than using glibc 231 versus 230, for example. So,
okay, so
this is what we definitely looked at.
what I'm hearing is that with the current solutions, we can maybe put the software reproducibility issue, maybe push it down a bit as being a critical issue. So we're back to the result reproducibility and
I would say it helps.
issue.
It helps a fair bit.
Yeah.
Yes, exactly.
Okay,
It's not perfect, but that's why is it good enough? And for us, it is
Tui.
currently. Yes.
Can I just quickly add to Stephen's question? I think the things that we can take from psychometric validity and validation and bring over some of those approaches, kind of broadly speaking, into every result. The bioinformatics community went into independent data sets, which is almost like pre-registration in some ways, right? That you had to show the same result holds in another data set. And I think if we can approach it as a field to bring all of those components together for every paper out there, and this is going to mean less papers, which I think is okay, we might get to a state of better generalizability of this. I think that's what we are looking for. It's not reproducibility, it's generalizability of the knowledge that we expect.
My sense of my bit anxiety is if I read the paper, there's only one pipeline that has been used. If I have no idea of the variability that would occur by taking other similar reusable tools, I have no idea whether that result is actually, like overfitting with that specific tool. And that's my kind of a general thinking when I read a paper, when I see a paper, like, how do we assess, what's that, what that, and hopefully most of the results will be kind of similar. There's definitely results that will go away. And not knowing and how much of those is to me a little bit of a, kind of, okay, we need to do something. But
So maybe I'll intercede here on one point that if I don't want to sound too much like the half full glass person, which I tend to be, right? But in science, we don't have problems with reproducibility. It's just a matter of time in most cases, right? And again, the case of the regressing out the global signal it's a good example. And time is important, right? You don't want to delay a field by 20 years because you didn't detect a bias and you could have detected a bias. Just want to make sure that it's clear that we have a scientific method that makes us, look over time at the results that stick on the wall, right? That other groups replicate, that other pipeline replicates, and that's really the fundamental method that we're using here. Now, at the faster times, we would like to run maybe a couple of pipelines to make sure that, results don't depend, that they're not biased, right, by the pipeline you're choosing. Right? Just to make, there's a lot of young people in the room, and yeah. But science is not broken. None of these methods is really broken. They all tend to get to a good point, and some of them might have biases, but you want to be better than that. You want to detect as early as possible potential biases maybe in a result that is primarily due to a library or a pipeline or a tool and not the others.
Well, it's the size of the bias.
Yes.
And since it's actually unknown in any given individual result, that is actually at least something you should think about.
Yes. Coming from Texas, I stand by that. It's the size. Yeah. There's a question there.
And the question I have is in terms of reproducibility. There is, even for the same type of experiment, there is some differences between how one group runs a specific MRI scan versus another group. So as a community, how can we standardize some of these methods so that it's not just the software and the other things that's downstream pipeline, but also from the experimental paradigm? How can we standardize this method so we can increase the robustness of our analysis and reproducibility part?
I can say a word on that. First of all, developing standards wherever whatever the goal is, if it's a standard for acquisition and everything, is always a difficult task. So usually for acquisition at least, a big consortium or big projects have proposed some specific acquisition that are kind of standard and been taken, like the HCP, the ADNI, that are so those things tend to be reused. At the same time, you've got the physicist on the scanner who says, okay, my scanner can do so much better. Why would I, curb myself and and limit the results and limit the? So I don't think we can, entirely do that standardization, which will be probably based on the most common scanners or the most the average scanner. So it has to be there's going to be variability. The question is, how do we consolidate results through meta-analysis, through across all those things, and that description of the results and of things has to be fairly deep to be able to actually know when we can consolidate things across cohorts, across scanners, across methods. So I don't know if that's an answer, but this is what it's prompted me.
I would say as much as possible, try to reuse what is out there. So when you do your stuff, share it, and then if there's something you're going to do. there are limitations. I'm thinking of the NIH toolbox that was used in the Human Connectome project, and I remember the people who used it, they were told by NIH, you got to use these instruments, but the various specialists are like, well, that's an awful one. Like, I wouldn't have chosen that. And so I think, given time, they did add some, but this is always a trade-off, right? There is the standard thing, but any standard has limitations, and then you have to deal with that.
So another way to say is that variability is also important. So standardization has the risk of limiting that variability, especially if standardization is adopted early during a field growth, the evolution of a field can be very risky, right? Some of these differences in toolboxes and tools and approaches that we have seen over time have effectively improved the field, because we have noticed that things had less bias or more bias by doing one way or the other one. So standardization is good. We're all working on the standardization here, but it has to be standardization on the point that you're working on something mature. You're standardizing something mature.
Are we out of time?
Yeah, how are we doing? So, okay, the moderator here. So I have another question, and then maybe we
No, the opening is in a minute.
Oh, we have to go?
Yeah, we have to.
Okay. So we're done.
Thank
Thank you.